Okay, so it's just a few minutes past 1, so I think it's a good time to start. There might be a few more people to join as we start the course today, but we want to make sure that we have time to go through everything. So as I said, welcome back to the Ensembl Genome Browser Workshop. This is our final day together, and Louise and I will be going through the final modules, looking at some of the different data types that we have available in the Ensembl Genome Browser. So I just want to quickly remind you of the structure for the workshop that we have today for the rest of today. So each of the workshop contains a number of modules, and each of the modules has a particular structure. So we'll start off with a presentation, just explaining to you about what the tool is or what the data is that we're presenting. And then we'll show you and talk to you about how Ensembl produces or processes the data. Then we'll move on into a hands-on demonstration. What I'll do, or Louise later on today will do, is show you how to use the tool or to get the data using the Ensembl Genome Browser. And this is a really good opportunity for you to follow along if you want to. Of course, if you don't want to follow along, you can just see what we're doing on the screen and then try and obviously replicate the steps later on in the exercises. So the final section of each module is the exercises, and these allow you to try things out for yourself and to go beyond the demo. So as we saw yesterday, we have a number of different exercises relating to each module, and the exercises, well, they mainly focus on human and other model organisms, but they are really just a starting point that allow you to begin exploring Ensembl for yourself as well. So please obviously look at the exercises and have a go at them. There are answers that you can check back to see how you're doing with if you get stuck. But equally, if you have your own species of interest or your own gene of interest, then please feel free to use the exercise session to begin exploring for yourself. Obviously, you can ask questions at any point during the presentations and the demos as well. But the exercises, Louise and I will be here to help you if you get stuck or to answer any other questions that you have. So all of the materials can be found through the Ensembl training pages. So we have the same link for the materials as we used yesterday. So I'm just going to quickly remind you how to find the materials. If you jump out of the, if you jump into a new tab in your internet browser and type training.ensembl.org, that will take you to the Ensembl training portal. So this is not the Ensembl browser itself, but this is our training pages with all the course materials. So you can click training materials in the blue bar at the top, and that will take you to our calendar of events. So here you can see our upcoming event for yesterday and today is for the DKFZ workshop. So you can click on that link and that takes you to our event. So this is where you have all the materials. Presentation slides that you used yesterday, which is where you can find out information about the course, a bit of background information, but it also has a space at the bottom for asking questions where you can include screenshots and links as well. So the idea of the living document is that it's a shared document that everyone has access to, and it will be available in the future as well. So you can always come back here and use the living document almost as a knowledge bank of the questions that you asked and the answers that we gave. At the very bottom, you can see a section called Demos and Exercises. We've obviously moved through some of these modules already, but you can see that we have exercises and demos for the variation, the relation, and BioMart modules, which are the modules that we're going to be doing today. So to access the demos and exercises, you just click on the module name, and then you can open either the demo, which contains the screenshots of the steps that we're going to demo, or you can the exercises where you can find the worked answers. So you've got the answer written out, a step-by-step guide, and then also a video showing you as well how to retrieve the data, the answer. Good. So what I'll quickly do is I'm going to put this link in the chat box so that everyone can access those materials again. Hopefully you have them already. And as I said, if you do get stuck on the exercises or if you have any questions, please do use the Zoom chat box for some short technical questions, or if you want to get a quick answer, then Louise will be on hand while I'm presenting, for example, to try and answer your questions as they go along. I'll also try and keep an eye on the chat box as well in case there's a question that I can answer in the course of my presentations. You can also, as I've mentioned, use the living document to ask a bit the longer questions that might need a bit of a discussion around them, for example. So this is the agenda. You can see that we've already gone through a number of these modules already. So we've done our introduction to Ensembl, and we've looked at genomic locations using the region in detail view. And then yesterday, just at the end of the workshop, we looked at the genes and transcripts with Louise. And then I quickly talked you through the comparative genomics data that we have in Ensembl. So today, these are our modules that we're going to be covering. So we're going to look at variation data in Ensembl and how we can use the variant effect predictor tool to get and to analyze our own variation data sets. Then we'll move on to having a look at the regulation data. So we'll be having a look at how Ensembl annotates regulatory features such as promoters and enhancers along the genome. And then Louise is going to be finishing by showing you the BioMart data export tool. It's a really useful tool for exporting data sets from Ensembl. So I think that will be quite a nice finish. And then we'll wrap everything up just before 5 o'clock German time with some final information about how you can continue using Ensembl in the future. So let's move on to our first module for today. We're going to be looking at variation data. So what I want to do in this module is to talk to you about how we can classify variants. And then thinking specifically about Ensembl, we want to think about the different species that have variation data available for them in Ensembl, and the sources of all the variation data that we display in the browser. So as Louise mentioned yesterday, Ensembl is not a sequencing project. So we don't do any sequencing to either sequence the genomes of species or to identify variants. So we rely on different research groups and collaborations and consortium around the world to deposit variants into variation data archives, which we then obviously import into Ensembl and analyze and display in our browser. So in the demo, we're going to see how we can explore variation data in a number of different contexts. So we have the gene tab, which you saw yesterday. We're going to see how we can look at variants within a gene. Then we're going to look at the location tab. Again, you saw that yesterday. We're going to see how you can add variation data tracks to the location view. And then we're going to have a look at the variant tab. So the variant tab is somewhere we haven't been yet before, and it's a place where we can explore individual variants one at a time and find out more information about them. And then in the second part of the module, we're going to be looking at the variant effect predictor. So the first part of the module is going to be thinking about how we can look at variants in the Ensembl browser that are already known and have been identified. The variant effect predictor is a slightly different tool because it allows us to begin analyzing our own variation data sets. So if you've done your own sequencing or you have a list of variants that you're interested in, you can use the variant effect predictor to begin analyzing them and getting information about them. So we can think of variation data as coming in two main categories. So we can think of small scale variants that might include things like small insertions or deletions, or also what some people call SNPs, SNPs, single nucleotide polymorphisms, but more commonly these days are called SNVs, single nucleotide variants. So this is a very simple example here of what I mean by the insertions and deletions and SNVs. So you've got here, for example, maybe the reference sequence is this green individual here, and then the blue is a sequence of a second individual. And you can see here a deletion of a C. Here they have an insertion of a G, a single base pair. And then here we have what we would call a SNP or an SNV. So it's where there's no insertion or deletion, but just where the base is changed at a specific position. Then we have things like, then we have large scale variants. So these are variants that will affect maybe hundreds or thousands of base pairs at a time. And they can include things like large deletions or insertions, but also duplications and translocations as well. So in Ensembl, there are 24 different species with variation data available. So human is the most well-studied species on the planet. And you can see that is reflected in the number of variants that we've identified across all of the humans that have been sequenced. So in Ensembl, we have 680 million variants in the Ensembl genome browser with information about them. But then there are obviously lots of other species that have also got lots of variants identified as well. So mouse, very important model organism, has 84 million variants in the Ensembl database. And then all of the other agricultural species, for example, have also got a lot of variation data. For people who are studying non-vertebrate species, there are also a large number of other plants and fungi that have variation data available for them as well. So where does the data come from? So there's a number of different steps that we have in Ensembl when we import variation data. So the first step is to import the data itself. Then we apply some quality control to check that the data matches up to what we're expecting. Then the step 3 is to look for associated data. So to look in other databases to see if there's additional information about the variants that we import as well. And then the step 4 is to apply our own analyses to these variants. So step 1 is the import of the data. So as I said, Ensembl is not a sequencing project. So we look to a number of different variation data archives that are available, and we import the variation data from there. So the most famous of these probably is called dbSNP. It's a database hosted by NCBI over in the States. And it's a database of short genetic variants for human. So we import human variation data from dbSNP. EVA, the European Variation Archive, is very similar to dbSNP. It has information about non-human species. So we import non-human variation data from EVA. Then we have DGV Archive, which has information about structural variants, large-scale structural variants. These databases generally have information about germline mutations, germline variants, sorry. But we can also think about somatic variants. So there's a couple of databases that are quite famous again that have information about somatic variants. So COSMIC is the catalogue of somatic mutations in cancer. And we import variation data from COSMIC and the cancer gene census that have information about somatic variants and their associations with cancer. So the next step, as I said, is quality control. So we do a number of different things when we're looking at the variants that we've imported. So the first thing that we can do is check that they are associated with the correct genome assembly. So we can map from one reference genome assembly to the other. We also perform checks on the alleles to see that the reference matches up with the reference genome, the reference allele in the genome sequence. And we look for things, different particular flags that might indicate that those variants might be suspect as well. Step 3 is to look for linked or associated data. So for all the variants in step 1, we look in a number of other databases to see if there's additional information that can obviously give us that we can add in about the Ensembl variants. So we look for things like allele frequencies, phenotype and disease associations, and publication data as well. So this is just a snapshot of some of the databases, for example. So we might look to databases such as the 1000 Genomes Project to look for allele frequencies in different populations around the world. Phenotypes and diseases we often get from ClinVar, which again is a database hosted by NCBI that has information about clinical phenotypes and associations. And publication data, we look to places like PubMed and Europe PMC. So the first type of linked data that I said that we look for is the allele frequencies. So there's a number of different projects that have got allele frequencies for variants in different populations around the world, and this is the main set of projects that imports variation allele frequencies from. So we've got the 1000 Genomes Project that looked at healthy individuals from 26 different populations around the world to identify a catalogue of genetic mutation around the world. Then we also import allele frequency data from Nomad, which has mixed disease and healthy allele frequency data. Then we also import the same sorts of data from UK 10K project, TopMed, and the NCBI Alpha project as well, the allele frequency aggregator. The second type of linked data that we look for is the phenotype and diseases. So again, this is just a snapshot of some of the variation, the phenotype and disease databases that we look to. So ClinVar is, as I said, a database that has information about phenotype and disease associations, clinical disease associations. Then we also look to databases like OMIM, Decipher, DDD as well. There are also some specific, some species-specific databases. So we look to IMPC for mouse phenotype data, and RGD for rat, for example. The final database that I wanted to mention specifically is the GWAS catalog. So this is a project hosted together by the NHGRI and the EBI here in the UK. And it's a catalogue of all of the different genome-wide association studies that have been published. So all of the different phenotype associations that have been identified in these GWAS publications are all compiled into this GWAS catalogue. And we have all of the associations from the GWAS catalogue in Ensembl as well. The final step, as I mentioned, is to apply our own analyses to these variants that we've imported into Ensembl. So the first thing that we do is we predict the variant consequences, which we're going to think about in just a moment. We also look to see if we can calculate any effects that these variants are going to have on the protein structure and function. We also calculate linkage to equilibrium, and we look at the variation conservation across species by performing some alignments as well. So as I said, the first thing we do is we calculate the consequence of each variant. So where a variant falls in the genome will have an effect, well, it is going to have a downstream effect perhaps on the gene and the protein structure and function. So we can think about variants in terms of the position that they fall in the context of the genes in that region of the genome. So a variant that falls in a 5' upstream or a 3' downstream region of the genome might not have a direct effect on the gene and the protein structure and function, but it could fall in a regulatory region, for example. So that could affect the expression levels of genes in that region. Then we can think about another variant, for example, that might fall within a gene, but not in the coding region. So it could fall in a UTR region, or in an intron, or in a splice site. Again, these variants won't have a direct effect on the protein structure and function, but it could affect the transcriptional, the transcriptional level. So it could affect the mRNA stability. It could affect the protein production from the mRNA. So all of these variants have the potential to have an effect on the cell through altering expression levels of the gene, or affecting splicing, for example. Then we also have variants that might fall within the coding regions of genes. So we can think of the coding variants as coming in two main categories. So we can have a synonymous coding variant, and a synonymous coding variant is where you have a change in the genome that does not result in a change in the protein encoded at that position. So we know that the genetic code is degenerate, so we can have more than one more than one codon encoding for the same amino acid. So what that means is that we can have a change in the genome, but that does not result in a change in the protein. Equally, on the other hand, we can also have non-synonymous coding variants. So this is where we have a change in the genome that does result in a change in the protein or amino acid encoded at that particular position. So this is what we call a non-synonymous or a missense variant. So this is what Ensembl does. We take all of the variants that we import in step 1, and we map them on the genome to see where they fall in the context of the gene, and we assign what we call a sequence ontology term. So these are the consequence descriptions. So just how Louise yesterday described the gene ontology, which was a standardized vocabulary of terms, that describe gene function. Sequence ontology is a standardized vocabulary of terms that describes the sequence-based changes. So I don't expect you to be able to read or remember this whole table, but the idea is that we have these different sequence ontology terms that you can see here in the left-hand column that are matched up with a description and a specific sequence ontology ID. So any variant, for example, that is a non-synonymous missense variant, so those non-synonymous coding variants, these are going to be labeled as missense variants. So if you look in the table here, just this one example here, a missense variant is described as a sequence variant that changes one or more bases, resulting in a different amino acid sequence, but where the length is preserved. So this is the definition of a non-synonymous coding variant. And so every variant that satisfies this definition will be tagged with this missense variant term, and it will be color coordinated with these colors that you can see on the left-hand side as well. So if we think about missense variants, those non-synonymous coding variants, we can also think of them in terms of their pathogenicity. So we can score the different variants in terms of how deleterious that variant will be on the protein structure and function. So there are various different algorithms that have been used to score the pathogenicity of the missense variants. So they're all very similar in the way that they look at similar types of data. So they look at, for example, how well conserved a particular protein is. They look at the chemical change in the amino acid substitution. So maybe you're losing a hydrophobic group or changing the charge on a particular amino acid when you have the variant and the amino acid substitution. And then they also look at the 3D structure and domains. So we're going to think about them in a minute, the different algorithms, but it's important to remember that they're all predictions. So they're all in silico predictions based on these sorts of considerations. So they're not facts, and they don't always necessarily agree with one another. So when you're looking at this data, it's always good to remember that this is obviously a good indication of what might be happening in vivo, but it's always good to experimentally verify these things and to corroborate with other evidence as well. So these are some of the different pathogenicity predictions that we have in Ensembl. So we have SIFT, which is works on a scale of 0 to 1. So when SIFT assigns a event a score of 0, it means that it's predicted to be deleterious, whereas 1 is more tolerated. Then we have polyphen, which is the inverse. So that also works on a scale of 0 to 1, but 0 is more benign and 1 is predicted to be more damaging. Then we have CAD, which works on a scale of 0 to 100. And again, 0 is more benign, with 100 being more deleterious. And then we also have REVEL, Meta-LR, and Mutation Assessor. So again, these all work on a scale of 0 to 1, with 0 predicted to be more benign or tolerated, and 1 is more damaging or disease-causing. Oh, the final thing to mention with the pathogenicity predictions is that we have all of this data in Ensembl with the scores and the color coordinations, and I'll show you how to view that in the browser in a moment. The final thing that I wanted to mention is the definition that we have around the different alleles of variants. So I just want to remind you of the what Louise was telling you at the very first presentation in yesterday's workshop. So when we're thinking about human, we have a single haploid reference genome that's meant to represent everyone on the planet. So obviously, we know that the human genome is what we think of as a hybrid or a composite genome, where the different contigs are the actual genome sequence that have been sequenced from different individuals. So we can take a region of the genome, one contig, for example, and we can see that this is the contig that might be sequenced from a lady. Maybe she's got a particular ethnicity or ancestry, so she might be European, for example. And you can see that she has this sequence in the part of her contig and a T at this particular position that I've highlighted in red. However, this lady is not particularly representative of the global population. So the T that this lady has is only found in 5% of people around the world. Everyone else has a G, so 95% of people have a G at this position. And G is the allele that we find in all primates as well. So if we align this region of the genome to orangutan and chimpanzee and gorilla, for example, the other great apes, we would see that G is also the allele that we find in this position. So that G is the ancestral allele. And the T that lady has does have some underlying disease susceptibility associations as well. So she might have been healthy at the time when she donated her DNA to the sequencing project, but she might have had an association to a particular to a particular disease later on in life. However, she was the lady who donated her genetic material to the Human Genome Project. So T is the allele that was sequenced in from her genome. So T is the allele in the reference genome sequence, and G is the alternate allele. So this is a very strict definition that we have here. So whatever allele is in the reference genome sequence is the reference allele, and then whatever is found in other sequencing projects afterwards, these are the alternate alleles. So in Ensembl, we'll always label this as T/G, so reference/alternate. So it doesn't matter how frequently observed the allele is, it doesn't matter if it has disease associations, whatever is found in the reference genome sequence is the reference allele. In many cases, you will see that the reference allele is the most commonly observed allele in populations around the world, but this isn't necessarily the case. So obviously my example here that I've used, I've made up to sort of demonstrate the point that the reference allele is not necessarily the most commonly observed allele. Okay, so what I want to do now is move on to the demo. So I'm going to show you how you can access all of this data in Ensembl. I've been keeping my eye on the chat box, but I don't think there are any questions at the moment. Before we start the demo, are there any questions that came up from the presentation? I'm happy for you to put them in the chat box or the living doc, or you can even unmute yourself and ask if you have any questions. Great. Okay, so if there are no questions, I think we'll start with the demo. You can always put the questions in the chat box as we're going through as well anyway. So what I'm going to do is just jump out of my presentation. And we're going to search, we're going to go to Ensembl for this gene called MCM6. So I'm going to go to the Ensembl genome browser, just like we did yesterday. And I'm going to search for the gene MCM6. I'll put this in the chat box for you as well, in case you need to copy that to follow along. Okay, so it looks like, well, we did report this issue to the web team yesterday, but hopefully, well, the website should be working better than it was yesterday. If you are having problems, or if it's going too slowly for you, then remember the mirrors that we spoke about yesterday. You can use the US West, the US East, or the Asia mirror as well. So it looks like the page is loading for me. We'll see how quickly the different pages load. So the first thing we have to do is navigate to the Gene tab, and we can do that by clicking on the link in the search results to navigate to the MCM6 Gene tab. So this is a place that you saw yesterday. Over in the menu on the left hand, actually, you can see this section called Genetic Variation. So we haven't explored this yet. So what I want to do is firstly click on Variant Table in the menu on the left hand side. So what you'll see on this page is a table, a very, very large table that has information about all of the variants that are located within the MCM6 gene. So there's a number of things you can do with this table, but the first thing that you might want to do is to export it. So I'm going to show you how you can filter and search this table in the browser. But if you want to just export all the data to begin processing and analyzing it, you can use this Excel icon in the top right-hand corner. So if you want to export the data, that's okay, but you can also begin filtering the data in the browser. So you can use the search box here, or you can use these filters in these blue buttons at the top here. So you can filter based on the global minor allele frequency. You can filter based on the SIFT or the polyphen score, or you can filter on the consequence, or on any of the other columns as well, actually. But we're going to use the consequence filter. So as you can see at the moment, all of the consequences are turned on. So we're seeing all of the variants that fall within the MCM6 gene. But if we just want to see the missense variants by themselves, we can click this blue button in the top left-hand corner that says turn all off. And then we can click back onto missense variant just to turn those variants with that consequence on specifically. So then we can click apply. And that will filter our table to only show us the missense variants in the MCM6 gene. So I just want to spend a couple of minutes just going through the table and showing you what we have in this table. So in the left-hand column, you can see we have this variant ID. So this is a unique identifier for the variant. These are clickable, so you can click on them to navigate to the variant tab. We're going to explore that in just a moment, but so you can navigate to the variant tab this way by clicking on the IDs in the table. Then you can see we have the location, so the chromosomal coordinates. Then we have the alleles. So you can see we have T/C. So that means that T is the reference allele and C is the alternate. So what that means is at this position in the genome that you can see here, the reference allele is a T, but a C has also been identified. And we're going to see what happens when we substitute the T for a C. So the next column is called global minor allele frequency. And if you hover your mouse over the header, the column title, you can see that the global minor allele frequencies are derived from the 1000 Genomes Project. So if a variant was not identified in the 1000 Genomes Project, it won't have a frequency here in this column. Then we've got the class, so this type of variant that we have here, it's a SNP, and we can see the source. So the source in this case is the dbSNP. It is for most of the variants you can see here, dbSNP. The next column is called evidence. So here we have different icons that tell us about the different data and the projects that this variant was identified in. So you can see this variant at the top here has frequency data available, and it was identified in the NOMAD project. If we scroll down, we can see this variant, for example, has frequency data. It was identified in the 1000 Genomes project. It's got citation information, but it was also identified in the ESP, the EXACT project, the NOMAD project, TopMed project as well. So this is just telling you about data and projects that the variant is associated with. The clinical significance comes from ClinVar, so you can see if a variant has any clinical associations from ClinVar. The consequence type tells us well, in this case, it's always missense because that's what we filtered, but you could hover your mouse over the definitions as well to see the description. Then we've got the amino acid change here. So here we've got the glutamate changing to a glycine when we change a T to a C, and this is at amino acid position 820. Then we've got these different pathogenicity predictions. So you can see SIFT, polyphen, CAD, REVEL, META-LR, and mutation assessor. So if you hover your mouse, you can see the description of the score, what that means. And you can see as well that, some of them are agreeing with one another, you do not agree with each other. So this is why you should always use these as a starting point, but then look in more detail at other evidence that you have available to you. Good. Okay. So what I want to do now is show you how you can access data through the location tab. So at the moment we've been in the gene tab. You can see that's what we searched for at the beginning, the MCM6 gene. But in the blue bar at the top, we also have this location tab available to us. So we can click on location. Again, we saw the location tab yesterday. This is the location where the MCM6 gene is located. So when we search for the MCM6 gene, this location tab automatically opened with the genomic region where the MCM6 gene is found. And these are the tracks that we have all turned on by default. So what I'm going to do is go back to this blue button on the left-hand side, configure this page. I can click here to find the variation data tracks that I can add to this view. So just like we did yesterday, we click on configure this page and we open up this pop-up window. And you can see all the tracks that are turned on, and then you can also turn on all of the other tracks that you need to as well. So we have in the menu on the left-hand side, this option, this category called variation. So if we click on variation in the menu on the left, you'll see all of the variation data tracks that you have available. So you might not have the same configuration as me. I've been working with Ensembl this morning. So, but you can see that you have these tracks that are turned off where the boxes are white, empty, and then you have some tracks that are turned on. So to turn on a track, all you have to do is to click the box. So for example, we have these tracks associated with 1,000 genomes data. So maybe I can turn off the tracks relating to the African populations, and I'll turn on the tracks relating to European populations. So you can see here, these are the continents for the genome projections, the American populations, the Asian populations, the European populations. So each continental population has two tracks. Each track is going to show us all of the variants that were identified in the European individuals. And then this second track is going to show us only the variants that were identified at a frequency of at least 1%, so the common variants in the European populations. As we scroll down, you can see that we have other tracks that are available to us. So we can find tracks that show us variants that are associated with phenotypes and diseases. So already by default, we have turned on this track called all phenotype associated variants. So any variant that's associated with the phenotype will be shown in this track here. Then we have other tracks for LSDB associated variants. We have phenotype associated variants, phenotype annotation, Then we have arrays, we have copy number variant probes, and we have a whole set of tracks for structural variants as well. So obviously we're not going to have time to look through all of these tracks, but the idea is for you in the exercises and in your own time to explore these different tracks and see which tracks are going to show you the data that you need for your studies. So when you're happy, you can click on the tick in the top right-hand corner to save and close. And my page is just reloading at the moment. So you can see that as well as the usual tracks that we can see, the contigs track and the genes track, for example, with the MCM6 gene, we also have these variation data tracks at the bottom as well. So this variation data track is showing us the nomad variants. So that was turned on by default. But then we also have these tracks that show us the variants from the 1,000 genomes project, 1 kg, and then from the European populations. So we have, these are all the variants identified from the European populations. And then in the track underneath is a subset of the variants from the track above, but only the variants that are found at a frequency of at least 1%. So all of these variants are interactive. So all of the data on this page you can click on. So if you hover your mouse until it turns into this sort of pointed finger icon, you can click on each variant and get more information. So you can see that this variant, for example, you can see the alleles, you can see the global minor allele frequency, and you can see the consequence as well here. You can click on the link in this pop-up window to navigate to the variant tab, which is what we're going to do in just a moment anyway. So you can see all the variants are color coordinated. So if you look to the very bottom, you can see this section in the legend called the variant legend. And this shows you all the different colors that the variants might be colored. So we can see, for example, these dark blue variants, for example, these are intronic variants. And this makes perfect sense. So if we now look at these variants that are dark, when we map them to the transcripts of the MCM6 gene, you can see that they're falling in this intronic region of MCM6. So they are intronic variants by definition, by the sequence ontology definitions. Good. Okay. So the final thing that I want to show you before the exercises is the variant tab. So as I've said, you can navigate to the variant tab by clicking on the IDs, either in the location tab or in the variant table that we saw earlier. But equally, you can just search for a variant ID if you know of it, like from maybe the literature, or you already have a note of it. So what I'm going to do is use this search bar in the top right-hand corner, and I'm going to search for my ID. So it's, in this case, it's RS 4988235. And again, I'll put that in the chat box so that you can copy that if you want to follow along. And then you can press enter. And then in the search results, we just have a single result. We searched for a unique ID, so we can click on that search result. So this brings us to the variant tab. So here we can find out more information about this individual variant. So again, you can see that the variant tab in the blue bar at the top, and we have this menu on the left showing us all of the available pages for this variant. So at the moment, we're in a summary page. So you can see that this variant is an intronic variant. We don't know which gene from the summary page, but we can find that out in a moment. And then we can see we've got the alleles. So we've got G/A/C. So this is a triallelic variant. So we've got the G is the reference, and then the A and the C are both alternate alleles. You can see the location here, and you can also see a description of this variant in VCF format here. So this is a common variant notation format. You can also see the original source. So you can see that this variant was identified from DBSNP or imported from DBSNP. So you can click on the link here, and it will take you out to the DBSNP pages for this variant. So you can obviously see what's happened here, and you can cross-reference to what you see here with what you see in Ensembl. So from the variant tab, there's just a few places in the menu on the left-hand side that I want to explore. So the first one is near the top. It's called Genes and Regulation. So here we can find the effects and the genes and the regulatory features that are affected by this particular variant. So here we can see a table that shows us the gene and the transcript consequences. So firstly, you can see that our table has 6 rows. And this is because the gene where this variant falls, MCM6, has 3 transcripts, but we have 2 alternate alleles. So what we do is we calculate the consequence for each of the alternate alleles against each of the transcripts where the variant is located. So in that case, you can see we have a prediction for the A allele for this first transcript of MCM6, and then the C allele for the first transcript, and then the A allele for the second transcript and the C allele for the second transcript, and then finally the A allele for the third transcript and the C allele for the third transcript. Then you can just see that the consequences predicted for each of the alleles against each of the transcripts. So in this case, it's an intronic variant. So there's a question that's just come in on the chat box. I'm going to address this at the end of the demo, if that's okay, just because I think it will work quite nicely to answer it at the end. The next thing that I want to show you is the population genetics. So this is on the menu on the left-hand side. This is where we can get allele frequencies in different populations around the world. So if you click on population genetics, you should find a number of sections on this page for the different projects that have allele frequency data. So at the very top, we have 1000 Genomes Project. But if you scroll down, you can find information from Nomad Project, NCBI Alpha, and then also for this variant, TopMed, UK10K, and the Gambian Genome Variation Project as well. So let's use the 1000 Genomes Project as an example. So you can see that we have these pie charts that show us the percentages for the observation of the alleles in different populations. So when all the populations are combined, we observe the G allele at 84% and the A allele at 16%. If we have different continental populations, you can see in African populations, it's 97 for G and 3% for A. In European populations, it's more of a 50-50 split. And then in the East Asian populations, in fact, 100% of the alleles observed were a G. So this actually is quite a nice example showing you how the alleles might be observed at different frequencies in different populations around the world. So if you want more information about a particular continent, for example, the European populations, you can click on the subpopulations button underneath. And that will open up a whole new set of pie charts that show you the individual populations in this European continent. So we have the first population is the Utah residents, so in America, but with a Northern and Western European ancestry. Then we have Finnish, British, Iberian in Spain, and then Tuscany in Italy as well. And again, you can see that there's a difference in the allele frequencies observed in each of these populations. In the table underneath, you can see similar type of data, but represented in a tabular form. So you can see all the populations here in the column side. Our chart presenting the allele frequency observation. So E is found, as we said, at 84%, and A is found at 16%. Now, it's important to remember that the 1000 Genomes Project, it had many different facets to the project, but the main part of the project sequenced 2,504 individuals from 26 different populations. But obviously humans are diploid. So every person that was sequenced as part of the 1000 Genomes Project contributed 2 counts to the allele frequencies. So here we can see the raw counts in brackets here. So if we add 4,200 to 808, we should get 5,008, which is 2,504 times 2. Over on the right-hand side of the table, we can see the genotype frequency. So what we can also find from the 1000 Genomes Project is the frequency that the different genotypes are observed in different individuals. So you can see here, for example, that the G, people who were homozygous for G is 75%, roughly. Then we have homozygous for A is 8%, and then heterozygous age is around 16%. The final thing that you can do, if you want to pick a particular population, you can do that, or you can click on the link at the top here for all populations. If you click on genotypes, in the column on the right-hand side, it will jump you down to the bottom of the page. And it will show you the genotype of this particular variant position in the different individuals from the 1000 Genomes Project. So obviously we don't know who these individuals are. They just have these unique accession numbers. So you can tell that this female with this ID from the British population is homozygous for A at this position. So if you want to, you can go back to the 1000 Genomes Project. You can look at this particular individual. You can even order immortalized cell lines that have been derived from this individual, so that you know that you can perform perhaps cell culture assays in the context of a particular variant or particular genotype. Good. Okay. So just scrolling back up to the top of the page, the final thing that I want to show you is the phenotype data. So if you click on phenotype in the menu on the left-hand side, here we'll find a page with all of the phenotype and the disease associations that are linked to this variant. So here we can see the table. So there's a number of different phenotypes. So we've got blood protein levels, body mass index, gut microbiota composition. These are all associations that come from GWAS studies. So you can see the source is the GWAS catalog here, for example. Then at the bottom, you can see a final phenotype that comes from ClinVar. So this database of clinical associations. You can see here we have this column called supporting evidence, where we can click to get all of the linked publications that link this variant with this particular phenotype. And you can click on the external references as well. Good. So the final thing to point out is that you can click on these phenotypes. So if you click on lactase persistence from the table, it will take you to the phenotype tab. So this obviously is showing you, this is a new tab in the blue bar at the top. This is a table showing you all of the features, the variants and the genes that are associated with this particular phenotype. So you can see our variant that we searched for is in the table here, but then these are these other variants that are also associated with lactase persistence. So obviously what I've done is I've gone on a particular route through the browser. So we started off looking at a gene and the location, then we searched for a specific variant, and then we went to the variant tab and the location tab. But you could equally imagine doing that in the reverse direction. So if you're coming to Ensembl and you have a phenotype as your starting point, as you're interested in a particular disease, you can search for that disease from the homepage and navigate to the phenotype tab. And from there, you can begin exploring all of the genes and variants that are associated with your phenotype of interest. So just because I've shown you one way, it doesn't mean that you can't explore it in other directions as well. Good. So that brings me to the end of the demo. What I wanted to do is just answer that one question that came in on the chat box, and then I'll set you off on the exercises. So Roland has asked, if you have a variant you know is relevant and recurrent in an amino acid code, such as MyoDL122R, what is the easiest way to find the related data in Ensembl? So the variant that you've described here is what we call in what we call HGVS format. So this is a variant notation format that we don't support for searching from the main Ensembl website. However, you can find variants in HGVS format using the variant effect predictor. So what you can do is you can actually put in variation data in this format into the variant effect predictor, and it will find all of the variants that are in the Ensembl database, and it will give you the associated IDs and all of the data that we've been looking at as well, which you can explore from that point. So I think the variant effect predictor in the next module will be quite interesting for you to use. I think that will answer your question. Good. Okay. So if there are no further questions, let's move on to the exercises. So all you need to do is go to the training pages and click on the variation module here. And you can find the exercises that are available for this section. So you've got an exercise looking at human population genetics and phenotype data, exploring a SNP in human, and then we also have an example with mouse, and then also if you're studying non-human species, then you might like to look at tomato as well, for example, or fungi. So I think it's just coming up to 2 o'clock there in Germany. So I think we'll have maybe 20 minutes to have a go at these exercises. So what we'll do is we'll come back together at 2:15, and we'll see how you got on with those exercises. But if you do have any questions, then please just message us in the chat box, and Louise and I will be there to help. Hi everyone. It's 2:15 now, so I just wanted to bring us all back together and see how we were getting on with the exercises. There was one question that came in on the chat box, which I think we've answered. So I don't know if there are any other questions that came up from the exercises, or if you got stuck on anything in particular. If there are no questions, could you just indicate that you're happy to move on by using the Zoom reaction? So just putting your hand up or using the thumbs up icon, just let me know that you are happy to move on to the next module. Perfect. So everyone's happy. There's just one question that's just come in from Sarah. So I'm just going to answer that and then we'll move on to the VEP module. So it says, why are there some RS at the same position and amino acid SNP but have a different RS number? This is a good question, Sarah. So maybe I'm not sure exactly of the specifics of what you've been looking at. So maybe if you, in the living document, if you could put some links to the pages that you're looking at and the IDs of the variants that you've identified where they're sort of maybe overlapping, then we can look into that in a bit more detail. But I'm sure there's a reasonable explanation, but I don't know it without looking. So yeah, put it in the living document, and Louise and I will take a look later on after the VEP section. Good. Okay. So what I want to do now, as I said, is move on to having a look at the variant effect predictor. So this first section of the variation module was looking at how we can export information about known variants from Ensembl. What we want to do in this second section is explore this tool that we have called the variant effect predictor. So this is a tool that you're able to use that will analyze your own variation data sets. So the idea is that you come to the Ensembl variant effect predictor with a list of variants. So you might have identified these variants in a number of different ways. So you might have performed your own sequencing experiments and then done some alignment and variant calling. Or you might have done some genotyping arrays, or you might have even just searched through the literature and have a list of SNPs. So Ensembl can't help you in this upstream process. We don't have any tools for doing any alignment or variant calling. But once you have your variants that you're interested in, you can use the variant effect predictor to begin analyzing them in more detail. So the idea is that you come to the VEP with your list of variants, and these can be variants in a number of different formats. So they might be a list of variants just given the coordinates and the alleles, or it could be variants in VCF format, which is a common format for variant callers as an output file format. It could be HGVS. So just as we had the question that came in earlier from Roland, if you've got variants in HGVS format, then you can use those as input in the variant effect predictor. Or you might even just have a list of variant IDs. So you might have a list of IDs, RS123, RS1234, etc., going down. You give all this variation data to the VEP, and then out of the other end, you can find the names and the IDs of the affected genes and transcription proteins and get the consequence predictions, the sequence ontology terms associated with that. If your variants are missense variants, you can also find out their pathogenicity. So you can get SIFT and polyphen scores, for example. What the VEP will also do is it will look to see if your variants are known or novel. So if your variants are, what the VEP will do is it will look through each of your variants one by one, and it will see if they match up with any of the variants that we already have stored in the Ensembl database. So if the variants are known, i.e. we have the information about that variant already stored, then you can get the frequency data, any literature citation information, phenotype associations, and those sorts of things. If your variant is novel, i.e. your variant that you've inputted doesn't match up with any variant in the Ensembl database, then it will still be processed and analyzed. You can still get data, but obviously there won't be any frequency data already associated with it, for example. And you can also get regulatory region consequences and splicing consequences as well. So just to remind you of the process, so this is taken from the slide that we had in the previous presentation. So what the VEP will do is it will take your variants one by one, and it will plot them in the genome based on their genomic coordinates, and it will give them a sequence ontology term based on fall in the context of the genes annotated in that region. So fall within the genes will determine the sequence ontology term. So if you want to use the VEP, you've got a number of different options. So firstly, we have a web interface. So this is obviously a nice point and click interface that doesn't require any technical or programming skills or knowledge. This is best for smaller data sets, perhaps. Then we also have a standalone Perl script, so which you need to install and run from the command line. So if you have larger data sets of maybe thousands or millions of variants, then you might want to consider using this command line option. I'm not a bioinformatician myself. I've obviously picked up things from working in Ensembl, but you don't need to have any skills in programming or coding to be able to run this script. You just need to know a few commands from the command line. So this could be an option for you if you have larger data sets that you want to begin analyzing. And then finally, we have the REST API, which does require some knowledge in programming and coding. So the method that you choose will depend firstly on your technical skills and knowledge that you have, and secondly, the data set size that you want to analyze. So it's best to choose the appropriate method depending on what you have available to you. So what we want to do is we're just going to use the web interface for our demo just to show you how the VEP works. We've identified 5 variants, so we're just going to have a small data set of 5 variants on human chromosome 9. So we have a C to an A at this position, and then a deletion of an A here at this coordinate, and then C to A C to G, and a G to A variant substitution as well. So we want to use the VEP to determine if the variants have already been identified in Ensembl, so if they're known or novel, what genes are affected by the variants, so the names and the IDs of the genes with the sequence ontology terms, and then if our genes affect any gene regulatory features as well. Okay, so if you want to follow along with me, the first thing that you have to do is get the data that we're going to use as input. Go to the training pages and navigate to the VEP module here that we have for this section. You can open up the VEP demo panel, and you'll find actually, if you scroll down a little bit, this is our input data in VCF format. So you can copy this and we'll be pasting it in at the right point in the demo. So if you want to follow along, please make sure you've come here and you've copied that data that we can paste in. So now you've got that data, you can go back to Ensembl, and you can actually see, even at the very top in the blue bar, we don't have to go back to the home page. You can just click on the link for VEP in the blue bar at the top. And this will take you to the page where you can see all of the information about the VEP. So you've got all the documentation for the web interface, the command line tool, and the REST API as well. So you can read all this documentation in your own time. If you want to run the VEP, the web interface, you can just click on the Launch VEP button here. So I've run some jobs before. So what you should find if you have not run a job before is your data input page like this. So we can walk through the different steps that we have here for providing our data and selecting our different options. So at the top, we have our species. So by default, we have human selected here, which is good for us. This is what we need. But obviously, you can change the species if you want to using the button here if you're studying a different species. It's important to notice as well here that we're using the GRCH38 human genome assembly. So that's, again, fine for us because the variants that we have are have coordinates that are relating to the GRCH38 genome assembly. If you have variants that were identified in the context of GRCH37, then what you'll have to do is go to the, you can't change the assembly here, you'll have to go to the archive, the GRCH37 archive, and run the VEP from that website instead. So we don't need to change anything for our query here. So we can just give our job a name. So I'm just going to call this one DKFZ. Then we have to give the VEP our data. So we've got a number of different options here. We can paste our data directly in the box, which is what we're going to do. Or you can upload a file. So if you have a file saved locally on your computer, you can choose a file to upload from your computer. There is a size limit on these files though. So if you have a really large file, you can place it into a public directory, like on an FTP server, and you can provide a URL. So this is really only necessary if you have really large files that exceed the upload limit here. So we're just going to use this text box because we have a small amount of data. Before we paste in anything, you can see actually, if you click on the examples beneath, you can see that the text box actually gets populated with some example data. So this is not our data, this is just some sample data that shows you the different formats that the VEP supports. So you can see we've got the Ensembl default formats. This is variation data in VCF formats, a list of variant identifiers, HGVS format, and SPEDI as well. So these are the different file formats that the VEP supports. So we can delete this. The VEP will automatically detect the data format that we're using, so we can just paste our data directly in the box. So as I said, this is VCF data, data in VCF format. So you can see that we have the first column, so we have 5 rows for our 5 variants, and the first column is the chromosome number. Then the next column is the coordinate, the genomic location. Then we've just given the variant a name here. So we've just called it variant 1 so that we can look at them in the output table. And then we have our allele. So here we have TA is the reference and T is the alternate. So this is the deletion of the A. Then we have the C to A, the C to G, the G to A, and the C to A. So these are our 5 variants. The next thing to do is to select our transcript database. Oh, I'm just going to close these so that you can see what I see as well. Okay. So the next thing to do is to choose our transcript database. So by default, we're choosing the Ensembl GenCo transcripts, which is what we're going to use in our example. But if you want to analyze your variants in the context of the NCBI RefSeq transcripts that Louise mentioned yesterday, so this is the gene and transcript set that is produced by the NCBI group over in the States, then you can use this as your reference data set as well. So this is just choosing which reference data set you want to use. So we'll use the Ensembl GenCo transcripts in this case. Then the next thing is just to pick our additional configuration. So we can open identifiers. You can see that some options are already selected for us by default. Someone's, okay. People are helping each other in the chat box. I like to see that. So yeah, there's just the variants that you can use. The identifiers section is allowing us to pick options that will return information about the genes and the transcripts and the proteins that are affected. So you can see by default, we're going to get the gene symbol and the transcript version ID. So the gene symbol is the gene name. So we're going to find the name of the gene and the ID of the affected transcripts. But you can also pick to see the ID of the affected protein and any UniProt IDs as well of affected proteins. The next thing to do is open variance and frequency data. So you can see that the first option is asking us if we want to find co-located known variants, and by default that says yes. So what the VEP will do is it will look at each of our variants and it will see if any of our variants are co-located, if they match with any known variants from the Ensembl database. So if they are co-located, if they are co-located, then we're going to retrieve this extra frequency and localization data. So we can get the 1000 genomes global minor allele frequency and the continental allele frequencies as well, if the variant has already been identified in the 1000 genomes project. We can also get the PubMed IDs as well for any variants that have literature citations. We have additional annotations. Again, some are selected by default, so we're going to find the biotype of the affected transcripts, we're going to find the transcript support level, the appriss ID, the appriss annotation, and the main annotation for any affected transcripts. But you can also select other options as well. So you can choose to find any affected protein domains. You can see by default, we're also going to get regulatory feature consequences. And you can also choose to get any phenotype associations as well. So there's obviously lots of different options here. I'm not going to go through every single one of them, but again, the onus is on you to go through and think about the different options and select the ones that are going to get the data that you need for your work. The next section is called predictions. So again, this is looking at pathogenicity and splicing predictions. So here you can see by default, we've got SIFT and polyphen. We're going to get the prediction and the score for both SIFT and polyphen, but you can also turn on other pathogenicity predictions as well. You can also get splicing predictions and conservation scores as well. And then the final two blocks are showing us the filtering and advanced options. So at the moment, there's no filtering selected by default. And I would really advise you to use no filtering at this stage. So what the filtering does, if you select it here, is it will actually filter your data before it gets displayed in the final output table. So you might actually end up losing interesting data. I think it's much more wise to actually process your full data set, not do any filtering at this stage. And then once you've got your full output table, then you can begin filtering and prioritizing and annotating from there. So I would say not to do any filtering at this stage, but you can do if you know exactly what you want to do and you know how to manipulate the data in the right way. And then the advanced options are really just for optimizing the running speed if you have large data sets. So we don't need to do anything here. Then the final thing to do is to click run. So what you can see here is a list of all of your jobs that you've submitted to the VEP. So you can see here, for example, last week I ran a couple of jobs on the VEP. So I've got these 2 jobs that are listed as done. But then this query that I have here, DKFZ, this one is currently queued. And now it's running. So the page refreshes every 10 seconds. So you should be able to see in just a few seconds that this turns from running to done. So hopefully you're also getting the same thing as well. From this page, you can do lots of things with your job. So you can use the icons here to save your job. You can edit and resubmit your job. You can share your results and you can delete your results. So if I don't want this one anymore, for example, I can just click delete and it will get rid of that for me. Good. So what you can do now is click on view results once your job is listed as done. And this will take you to the VEP results page. So at the top, you can see we have these summary statistics. So we have this table showing us that we've processed 5 variants. And of those 5 variants, 1 was novel and 4 are existing. So one of our variants has not been found in the Ensembl database. So that's a novel variant. And then 4 do match up with variants that are in the Ensembl database already. Our 5 variants have overlapped 4 different genes, which have in total 24 different transcripts. And we also have overlapped 2 regulatory features as well with our variants. Then these pie charts, these just give you a summary of the consequences for all of the variants combined. And then as we scroll down, we can begin to see our results table. So what we can actually see if we begin to scroll down is that we have lots of rows in this table. And they're not necessarily ordered at this point. So what I would advise you to do is click on this gray arrow in this for this column uploaded variant, and that will just order the table so that you have variant 1, the rows for variant 1 followed by the rows for variant 2, 3, 4, and 5 underneath. So what we can see if we begin looking at variant 1, you can see that we have variant 1 is an intronic variant. But it's an intronic variant of this transcript. We've got the transcript ID here of the COC4 gene. However, the COC4 gene has multiple transcripts. So that's why we have another row here, because variant 1 is also a downstream variant of this transcript of COC4. So what we actually have here is COC4 has got multiple transcripts, which might be different lengths. So this variant is falling in the intron of one of the transcripts, this one, but it's falling downstream of maybe this shorter transcript here. Then we have, again, another row. So this is it's an upstream variant this time of this transcript of COC4. And you can see all the rows for variant 1. So what we have in our variant effect predictor output table is a row for each affected transcript, each affected feature. If we scroll down, we can see that variant 2 is even a bit more interesting. So variant 2 is also affecting COC4. So it's an upstream variant of this transcript of COC4. It's an upstream variant of this transcript of COC4, but it's a synonymous coding variant of this transcript of TrueB2. So in this case, this variant has got multiple effects on different transcripts of different genes. So what we will see in a moment is that COC4 and TrueB2 are probably very closely located to one another. And this variant is affecting the transcripts of TrueB2, but it's also affecting the transcripts of COC4 as well. So obviously, because genes can be closely located to one another or even overlapping, a variant that falls right at this position here, at the junction between these two genes, can have effects on the transcripts of one gene and other effects on the transcripts of another gene. So we list them all in this table. Variant 2, interestingly, is also falling within a regulatory feature. So you can see here in a separate row that variant 2 is falling within this promoter. So we're going to think about the regulatory build later on today, but you can basically see that this variant is falling in this promoter region. And you can click on these IDs to go through and get more information if you want to. Variant 3, if we scroll down, is interesting for another reason. So variant 3 is a missense variant. It's also falling in the upstream region of TrueB2, but it's a missense variant of this COC4, of the transcripts of the COC4 gene. So if we begin to scroll across, we can actually see a lot of the different data that we have available to us now. So if we scroll across, we can see all of these different columns in our table. So variant 3, for example, is a missense variant that falls in these transcripts of COC4. It's falling in this exon 2. It's the cDNA position, the CDS position, the protein position, the amino acid change. So in this case, we're changing a serine to a cysteine, and that is encoded by the TCC that's being substituted for TGC. So this is the codon change here. The next column is called existing variant. So here you can see in this column an ID. So that means that this variant has got an ID already associated with it. So it's one of the known variants. If you scroll up, you'll be able to see that variant 1 actually has a blank column here for existing variants. So variant 1 is our novel variant, and the other variants that have IDs here, these are our known variants. Then we can see information for the transcripts. So we can see whether it's a main select transcript. We can see the APRIS annotation. We can see the IDs for the affected proteins, for UniProt, for example, as well. Again, you can click all of these links if you want to. Oh. Then you can see we've got SIFT and polyphen scores. So obviously not available for every variant, but if a variant is a missense variant, you'll have the SIFT and the polyphen score and prediction as well. You can see the affected protein domains if it falls in a protein here, for example. And just as we saw before, we have a variant that is, we have a verified in the 1000 Genomes project, so we can get the 1000 genomes frequencies here as well. The final thing to mention is, if we scroll further across, we can see any clinical significance. And over on the very right-hand side, we can see phenotype associations as well. So obviously variants might be directly associated with a phenotype, in which case you can see that here. But variants might also be associated, genes might also be associated with phenotype. So if a variant overlaps a gene that is associated with a phenotype, you'll also see this here as well. So you can see that this variant is associated with coenzyme Q10 deficiency, but it also it overlaps this gene here, which is the COX-4 gene, which is associated with neonatal encephalopomyopathy and respiratory distress syndrome. And you can, again, click on these links and get more information. Good. So that brings me to the end of the demo. I think there's not any questions to answer here directly. So what I want to do is give you some time to have a go at the exercises. And we're also going to take a break. So it's now 20 to 3. So what I propose we do is we spend 20 minutes doing the exercises for the VEP, which will take us to around 3 o'clock in Germany. And then what we'll do is we'll take a 20-minute break. So we'll start our next module again at 20 past 3. What that means is that actually you can spend the next 40 minutes however you want to. So if you want to take a break now and then come back and do the exercises afterwards, that's also fine. But if you prefer to start with the exercises and then take a break after a while, then that's completely up to you. So the next 40 minutes is up to you how you want to divide your time. As long as you make sure you're back here by 20 past 3, we'll be able to go on with the next module. So I'm not sure if there are any questions. I think there's one question that's just come in directly because there's some technical problems. So I'll answer that. But if there are no other questions, then please do go back to the training pages to find the exercises and run your own VEP queries. So there's one exercise looking at 3 variants in the CFTR gene, and then there's another exercise that's looking at a structural variant in VCF format, which you can copy and paste from here. Hello everyone, it's now 20 past 3, so hopefully you've had enough time to have a go at the VEP exercises, but also take a short break. I think it's time to move on to the next module, but before I do, I just wanted to see how you got on with the exercises. If there were any questions that came up about the variation data or the VEP, or if there's anything that was unclear or that you had trouble with. Good. Yeah, so there were a few questions that came in on the chat box and in the living document. So hopefully everyone's happy. What I think we'll do now is we'll move on to the next module, which is looking at the regulatory build in Ensembl, so the annotation of regulatory features. So there are, as we mentioned already earlier on in yesterday's sessions, there are 4 main types of data in Ensembl. So we have the gene annotation data, the comparative genomics data, the regulatory data, and now the 4th type of data that we want to explore is the regulatory data. So this is where Ensembl annotates the genome with functional regulatory elements such as promoters, enhancers, or repressors. So these are features that are able to control gene expression. So to annotate these features, we use a combination of different biological data types. So we use data that show us epigenetic marks, so histone modification and DNA methylation patterns. We also use ChIP-seq data that looks at transcription factor binding patterns and RNA polymerase binding. And we also look at the open and closed chromatin conformation that we assess through DNase I sensitivity assays. So the importance of gene regulation and the expression of genes and the levels of gene expression is very important. And we can exemplify this through the hematopoietic stem cell lineage. So if we think about all of the different blood cell types that we have, all of these cell types are actually derived from a single hematopoietic progenitor cell. All of the cell types in our blood, the differentiated cell types, are highly morphologically different. They've got very different functions. And this is because they express genes at different levels. So the genome in each of these cells is exactly the same, but it's the levels and the timing of the expression of the different genes in these cells that lead to the morphological and functional differences in these cells. So we say that these cells have different epigenomes, which is the epigenetic profile of the cell, expression profiles of the cells. So there are many different layers of transcriptional regulation that are at play within a cell to control the levels and the timing of the expressions of genes. So we can think of a number of examples just through this cartoon, for example, here. So this might be a region of we can see here just going off of the edge of the screen here, this is our coding region. Immediately upstream of a gene, you often find a promoter region. So this is a discrete region of the DNA that binds RNA polymerase, which is obviously the machinery that transcribes the DNA into mRNA. Within the promoter sequence, as well as binding RNA polymerase, we also find that a number of other proteins called transcription factors bind to specific sequences within the promoter region. So these are called transcription factor binding motifs, and the transcription factor proteins bind to these specific sequences, which then recruits the RNA polymerase to do the transcribing of the gene. What we also have are these other regulatory elements called enhancers. So enhancers are more distally located from the gene, but they have their own sets of transcription factor binding motifs, and they bind to their repertoire of proteins, and it's actually the 3D conformation of DNA bringing the proteins bound to the enhancers and the promoter together, which recruits the RNA polymerase, leading to transcriptional activation. You can also find that there are other elements such as repressors and insulators that bind their own sets of proteins that can actually modify and downregulate gene expression as well. So what we can actually see is that within the promoters and the enhancers and the other regulatory elements, as well as binding the proteins, well, the binding of the proteins to the transcription factor binding motifs can be regulated through a process called methylation. So within the DNA sequence, there are regions of CG-rich repeats that we call CPG islands. And there is an enzyme called DNA methyltransferase that moves along the genome and selectively modifies the cytosine bases within CPG islands with methyl groups. So there's a covalent bonding of the methyl groups to the cytosine bases within the DNA. And the methylation pattern in the promoters and in the enhancers can modulate the binding affinity of the transcription factors to the DNA. So perhaps methylation of a particular promoter might block the transcription factors from binding to the sequence in the DNA. So we already thought about DNA methylation as one level of transcriptional regulation, but the transcription can also be regulated by a number of other factors. So the conformation of the chromatin itself can lead to activation or deactivation of transcriptional activity. So in our textbooks, you can see here, for example, is normally wound in a very uniform and regular picture. So you can see, like here, it's a very nice representation of the regular compaction of the chromatin. However, in real life, what we actually see is that there are regions of chromatin that are very densely compacted, which is associated with transcriptional deactivation, whereas we also get regions of chromatin that are more open and less densely packed. And this is genes in these regions of open chromatin are associated with transcriptional activity. We also know that the nucleosome positioning can affect transcriptional regulation. And finally, we also know that the genes can be the expression of genes can be regulated by the modification of the histone proteins themselves. So in order to be condensed down into the chromatin and the chromosomal structure, the DNA strands themselves are wound around proteins called histones. So what we have here are these histone proteins. So there's 4 types of histone proteins: histone H2A, histone H2B, histone H3, and histone H4. And what we actually have is we have 4 different homodimers of these 4 proteins that come together that form the histone octamer, and the DNA is wound around these proteins to condense it into the chromatin and the chromosomal conformation. So these histone proteins, they're quite globular, but they have an N-terminal tail that extends away from the protein itself. And these N-terminal tails of the histone proteins can be post-translationally modified. So we can have methyl groups, acetyl groups, ubiquitin, sumoylation. All these different post-translational modifications can be added to these histones. And it's this pattern of histone modification that can also upregulate or downregulate the genes in that region of the genome. So we have all these different layers of transcriptional regulation, and we also have a number of different experimental techniques that can assess the pattern of these different regulatory mechanisms across the genome. So this is a diagram to try and summarize what we've just been thinking about here. So obviously we have our chromosome that's compacted in the chromatin that's compacted into the chromosome. So we have these regions of closed chromatin, and then these regions of open chromatin here that we'd think of as the hypersense. So these are regions of open chromatin that are sensitive to restriction by DNA's enzymes. So we've got DNA sequencing can assess the pattern of open and closed chromatin along the genome. We also can use techniques called FAIR-seq and ATTAC-seq as well to assess the open and closed chromatin pattern. We can use ChIP-seq to look at the pattern of protein binding along the genome. So we can use ChIP-seq firstly to look at the pattern of RNA polymerase or transcription factors binding to different regions of the genome. But we can also use ChIP-seq to look at the histone modification. So we can use antibodies that specifically bind to histone proteins that are modified in specific ways and look to see where in the genome those particular histone modifications are found. We can look at the pattern of methylation using a technique called bisulfite sequencing. So we can either have whole genome bisulfite sequencing or reduced representation bisulfite sequencing that assesses the pattern of methyl methylation along the genome. We can also look at the actual gene expression levels itself using RNA-seq data. And we look at we can use ClIP-seq and RIP-seq to look at the expression levels and the mRNA levels as well. So that's a very, very quick and basic introduction to epigenetics and the regulation of gene expression. So what I want to do now is think about how Ensembl could integrate this type of data to annotate regulatory features. So in step 1, we take all these different types of data that we've just been thinking about. We import them from various projects and different sources. Then in step 2, we process all of this data to predict the positions of the regulatory features, different types of regulatory features such as promoters, enhancers, and transcription factor binding sites. Then in step 3, we assess the activity of these features in different cell types. So for each of the cell types that we have available in step 1, we predict whether these regulatory features are active, poised, repressed, or inactive in each of the different cell types. And then we display all this data through the browser, and I'll show you how we can see that. So where does this data come from? So there's 3 main projects that Ensembl uses to get this type of data that we integrate into our annotation of regulatory features. So the first is called the ENCODE project. So the ENCODE project looked at 48 different human cell lines and 8 different mouse cell lines. And it generated ChIP-seq data for histone modifications, transcription factor binding, RNA polymerase, and open chromatin conformations using DNA sensitivity assays. Then we have the Roadmap Epigenomics project that looked at primary cells, again in the cultured cells, cultured primary cells, sorry. And then we also have the Blueprint project that generates similar data, ChIP-seq data and DNA sensitivity data, but this time for 20 different primary cells from the hematopoietic cell lineage. So what you might have noticed is that there's actually only data that we have now for human and mouse. So we have the ENCODE, the Roadmap, and the Blueprint project that have all generated data for different human cell lines. And then we have the ENCODE project that's generated data for mouse cell lines. So what you'll actually see in Ensembl is that we only have the regulatory annotation, the annotation of regulatory features for human and mouse. So these are the only two species that have regulatory data at the moment. We are hoping to expand this technology and this annotation to other species that have this sort of data available, but this is sort of in progress and something that we're working on. So step one is to plot all of this raw data from these different projects against the genome and to annotate the peaks of these features. So the blue bar here at the bottom represents the genome, and we align all of the data from these different projects to the genome. So you can see these what we call signals, which is the line moving along the genome, and wherever the signal increases in a particular region of the genome, that suggests that that particular modification or that particular experiment identified the modification in this region. So we can see here, for example, transcription factor A is predicted to bind in this region where the signal reaches a particular threshold. And we do the same for all these different evidence types. So we look at transcription factor B, also binding in the same region. Transcription factor C is binding up in this 5' region. So we do the same thing against the reference genome for all these different evidence types from the different projects. And wherever the signal passes a particular threshold, we label these blocks called peaks. So this peak here is where this red signal passes a particular threshold. And what you can actually see is in particular regions of the genome, there's different evidence types that are all clustered in the same region. So you can see here, for example, transcription factor B and C, histone modification 1, 2, and 3 are all co-located in this same area together. So what we do is we then look along the genome and we see if there are any other regions of the genome that have this exact same pattern, this exact same signature of evidence co-locating with one another. And if any of those positions are known promoters, then we infer that all of the other positions that have this exact same signature are also promoter regions. And we look for other patterns and we do the same thing. So we look for patterns and looking to see if there is any known promoter regions associated with that signature. The next thing we do is a process called segmentation. So this is where we categorize the different regulatory features based on the underlying signature. So this signature here, for example, of all of this underlying data might be associated with a promoter sequence. And then we have all of these two underlying evidence here in this particular region might be associated with an enhancer. So we have the segmentation of the regulatory features into different categories based on the underlying features. And then the next thing to do in step 3 is to predict the cell type activity. So the first thing to mention is that we do this process for every single cell line that we take from the different projects. And then we move along the genome in 200 base pair blocks, and we ask, in this 200 base pair block in any of our cell types, is there a regulatory feature annotated? And if the answer is yes, then we annotate a regulatory feature. And we move across sequentially along the genome in 200 base pair blocks. And if the answer is yes for any cell type, then we annotate that block there. So what we're building at the top here is what we call our multi-cell regulatory build. So this is the regulatory feature annotation for all of the cell types combined. And this is what you actually see in the browser. Then underneath that, what we then do is once we've collapsed all the cells into the multi-cell features, we then look at the cell types individually and we ask, in each cell type, are these features active, inactive, poised, or repressed? So if there's evidence to suggest that in cell type 1, for example, that this regulatory feature is active, then we will label that as active. And that suggests that transcription is actively occurring from that promoter. However, in cell type 2 and cell type 3, if there's no evidence to suggest that there's inactive, if there's no evidence to suggest that there's active transcription, then we might label them as either repressed or inactive, depending on the data and evidence that's there. And then we'll see all this in the genome browser. I'm going to show you how you can view this data in a moment. So one of the questions that I received yesterday in the chat box in the course was someone who was asking to look at the promoter and regulate and enhancer, so regulatory features for a specific gene. So it's important to remember when you're using Ensembl is that we do not link regulatory features to genes. So we have the genome assembly, we have our gene build, so the annotation of genes, and we have our regulatory build, the annotation of regulatory features. And you can see them side by side. So you can view them at the same time, but we do not explicitly say that this is the promoter of this gene. So you can obviously look at your gene of interest and you can look to see if there's a regulatory feature, a promoter in the 5' region. You can use things like cell line specific regulation data or tissue expression patterns, all these sorts of things to make that inference yourself. But that's not something that Ensembl explicitly does. So we're working again on the technology where we'll be able to make the direct associations between the genes and the regulatory features, but that's not something that we can do at the moment using the data. There's a few other types of regulatory data that we have in Ensembl. So for human and mouse, we have enhancers from the Vista project, and we also have mRNA targets from Tarbase. For human, we also have transcription start sites and enhancers from the Phantom project, and we also have EQTL data from the GTEx project. We've got methylation data from ENCODE, and we also perform some microarray probe mappings to the Ensembl transcript. So if you want to purchase a commercially available microarray to assess transcriptional, to assess gene expression, then you can look to find the microarray that maps to the transcripts of interest. As I mentioned, in the future, two of our main projects is firstly to apply this technology and this regulatory build to other species. So we're hoping to incorporate this for agricultural species from the FANG project, and then also to zebrafish from the Aquafang project. And then also we're hoping to use EQTL data and Hi-C data to be able to link regulatory features to genes. So what I want to do in this demo is have a look at a specific gene, and we're going to have a look at the regulatory features in the region of our gene of interest, pick one of the features, and then look at what we can find out about the activity levels in different cell types and the underlying data. So our gene of interest is called KPNA2. So if you want to follow along with me, you can jump out in your internet browser to the Ensembl genome browser. And then in the search box, search KPNA2. I'll just put that in the chat box for you. And then you can click go. Okay, so it's going slow again for me. So I'm just going to, oh, I was about to refresh the page, but it's just loaded. So from here, you click on the link for the human gene to go to the Gene tab. And then once the Gene tab is loaded, navigate directly to the Location tab by clicking on the Location tab in the blue bar at the top. So now the page is loaded. If you scroll to the very bottom and look at the region in detail view, you'll be able to see that we have our gene of interest. So this is our KPNA2 gene. It's got lots of different transcripts, so you can see them all annotated here. And you know that the gene is on the forward strand. So just as we discussed yesterday, we have the arrow for each of the next to each of the transcript names, indicating the direction of transcription. So this is at the 5' end of our gene. This is on the forward strand. So what we can do is we can look down and we can find this track called regulatory build. We can actually see that there's a promoter, which is this red. So you can see that the regulatory legend says red is a promoter. So this red feature, promoter feature, is just at the 5' end of our gene of interest. So this, again, as I said, we do not make the direct association between the genes and the regulatory features, but you can make a reasonable assumption that the promoter found at the 5' end of this KPNA2 gene is the promoter for this KPNA2. So to get more information, we can click on this regulatory feature. And in the pop-up window, you can see that we've just got information. It says it's a promoter. We've got the chromosomal coordinates, but you also have the link for the stable ID. So all of the regulatory features have a stable ID like the genes do. So these ones are ENSR for regulatory features. And then from the pop-up window, we can click on that stable ID. Again, things are going very slowly, so I'm just going to be patient. Hopefully things are loading for you, or you're able to use the mirror sites to find a server that's working a bit more quickly at the moment. So once this page is loaded, you'll be able to see a number of different things here. So at the moment, we're in the summary section. And if you're looking at summary, you can see the regulatory feature. You can see the Phantom 5 data, and you can see the mRNA targets from Tarbase here as well, annotated. Then we have this section called motif features. So these are the transcription factor binding motifs that are annotated within this promoter region. So the black motifs are experimentally verified, whereas the gray motifs are predicted, computationally predicted. So if you click on any of the motifs, for example, here, I'll try again just to, here we go. So this transcription factor motif that I've clicked on here has been experimentally verified in these different cell lines here, and it's the transcription factors TED4 and ERG that are binding to this transcription factor motif. You can look at the binding matrix by clicking on the link there. So this is the matrix, the binding matrix of that transcription factor binding motif, which you can download as PFM or as the SVG file. If you scroll to the very bottom of the page, you can see a table that shows us the different cells where the different cell lines where this transcription, where this regulatory feature, this promoter, is either active, inactive, poised, or repressed. So you can see that it's active in just the astrocyte cells in one cell line out of 118 cell lines that we have. It's inactive in 64 cell lines. It's poised in the 17 and actively repressed in the 36 cell lines. The final thing I want to show you is how we can look in more detail at the cells of our cell types of interest. So if you go to the top and click on details by cell type at the very top, so what we can see here, okay, so actually I've already, what I'm going to do is I'm just going to reset my configuration so you can see what I see. So this is the details by cell type page. And what we can actually do is we can begin selecting the evidence and the cell types that we want to view for this regulatory feature. So we firstly click on detail configure cell/tissue. And this will open up a pop-up window that allows us to select all of the evidence that all of the cells and evidence that we're interested in. So the first thing that we have to do is select our cell types of interest. So you can see that you can search the cell types using the search function here, but the cell types are also listed alphabetically. So I've selected the A673 cells and the astrocytes, but you can either click through and see all the different cell types that are available, or you can search for them using the filter function at the top there. So when you click on the cell types, as I've done here, you can see that on the right-hand side, we've now selected 2 out of 148 available cell types. The next thing to do is to click on the Experiments tab. And here we're able to choose all of the different experimental evidence that we want to view. So the experimental evidence is split into histones, open chromatin, and transcription factor categories. So if you click on histone, you can select the different histone modifications that you're interested in viewing. In this case, you can either deselect all or select all. So I'm selecting all of the histone modifications. You can also click on open chromatin. And again, there's just one type of evidence here, which is the DNA sensitivity, which we click on to turn that one on. And for transcription factors, you can see that we have 2 available. So depending on the cell lines that you've selected, there might be more or less data available. But for us, we've got the CTCF and EZH2 binding data. So when you're happy, you can click on the green Configure Track Display button in the bottom right-hand corner. And this will just allow you to select the formatting of the tracks that you see. So by default, you can see for each of the cell types, for all of the evidence types, we have this blocks and squiggle icon, like you can see here. So that tells me that I'm going to see both the signal and the peak, whereas I can change this to viewing only the peaks or only the signals by clicking on the button. So for us, we want to see all the data. So we're going to see the signal and the peak, but you can change it by clicking on the buttons here. When you're happy, you can click View Tracks. And then you can see the data as it's loaded on the page. So you can see that the data is organized by the cell types that we selected. So you can see we have the A673 cells at the top here, all color coordinated in this light blue color. And then we have the astrocytes underneath in this dark blue color. So for the A673 cells, we can see that this regulatory feature is sort of colored in this gray color. So if we click on it, we can see that the status is repressed. So that means that there's evidence that this regulatory feature is actively repressed in the cell type. So when we look here, we can look at this evidence underneath. So we have, these are the signals for the different cell type, for this cell type in different colors. And then these are the peaks that come from these blocks. So you can see that in this region, we have trimethylation of lysine 36 of histone 3, for example, in this region. We also have mono and trimethylation of lysine 4 of histone 3 as well. So these are the peaks from these underlying signals. So these histone modifications might be associated with transcriptional inactivity and repression. As we scroll down, we can see the astrocytes. So we can see that we have a large number of other data types. We have a large number of other histone modifications present in this region. So we also have, for example, methylation of lysine 20 of histone 4. We have this open chromatin. We have these other histone modifications. So these are all evidence that this regulatory feature is active in this cell type. So we click on the regulatory feature. And in the pop-up window, when it loads, we'll be able to see that this regulatory feature is active. So we can see active, and we can see all of the different motif features in this regulatory feature. Good. Okay, so that brings me to the end of the demo that I wanted to show you. So it's nearly 4 o'clock now. So what I want to do is give you 15 minutes to have a go at the exercises for the regulation section before I hand over to Louise for the final module of the day. So to do the exercises, you can again go back to the materials, open up the regulation section, and there are 2 exercises here. So again, both looking at human. So you can follow the instructions in the exercises to look at enhancers and other regulatory features as well in different regions of the genome. So I think, there's one question that's come in. I'm going to type an answer to this one because I'm going to have to do a bit of research, I think. But I'll type an answer to your question, Daniel, in the chat box. But for the rest of you, please do have a go at those exercises, and we'll come back together at 10 past 4 to begin our final module.
All right, so it's 10 past 4 now. Does anyone have any questions before I move on with the last module? Brilliant. If you do, just feel free to put it down in the chat box as always. Okay, so let us begin. Thanks for staying this long in the workshop. I'll be showing you BioMart, which is a tool with which you can download large amounts of Ensembl data with at once. And you don't need to know any kind of programming language at all. So I'm going to go through what BioMart is and how to use it, give you a brief introduction to the BioMart package on R, where you can find more information. And at the end, I will go through a demonstration and you'll again have time to do some exercises as well. Okay, so what is BioMart? It is a web-based tool to export Ensembl data without needing to have any programming skills. So you can build queries using a point and click method, so with your mouse, and you can generate custom data tables and files. But BioMart is not meant for downloading whole genomes, so you'll have to go to our FTP sites for this. And on BioMart, you can make up to 500 entries at a time. So if you're looking to do a query with more than 500 entries, I would suggest you divide it up into smaller queries so that BioMart can actually do this for you. And why should you use BioMart if you can get all the information you need from the different Ensembl pages? So you can do that. You can go to the different pages, so to the regulation, variation, all the gene annotation sites one by one, but it takes a lot of time, and it would be just like going to different shops for different groceries. So you'd have to go to one shop to get bread, to another shop to get cheese, and to another shop again to get your vegetables. So instead of making your life difficult, you can just go to a big supermarket, right? And get everything you need from this one shop. And this is what BioMart is. So it's a one-stop shop for all the different types of data you can get from Ensembl. On BioMart, you can query multiple genes or variants at once. You can convert IDs, you can extract loci, you can download multiple sequences in one go. So in other words, you can use BioMart for tasks that would be too time-consuming by going through the Ensembl browser alone. And you can find BioMart on Ensembl and Ensembl genomes. The structure of the browsers are all the same, as you know now. And the link to BioMart you can find on the navigation bar at the top of the page. And BioMart is available on Ensembl, which is dedicated for vertebrate genomes, but it is also available for Ensembl plants, metazoa, and for some species in protists and fungi. It's not, so BioMart is not available for bacteria because unfortunately, there are just too many of them. Okay, so now that you know what BioMart is and what you can get from the tool, I'll show you how you can make queries on BioMart. So the easiest way to use BioMart is to follow this 4-step principle. So step 1, you choose the data set and the database. So basically, you choose the data set. So there are 2 parts to this. You choose a database and a species. I will show you in the demonstration what I mean by that. The second step is to apply filters. So in this step, you narrow down your data set. Then step 3, which is specifying your output. So what you want to print on your table, all the details that you want in your results. again, it will make more sense when I do the demonstration in a minute. And then finally, in step 4, you choose the format of your results and how you want to export this. So to look a bit more closer to the different steps. So in step 1, we define the database that you want to do your search in. So there are different databases you can choose from. There is a genes, variation, and regulation database, and you can also look at certain mouse strains. And then finally, in the second step of step 1, you also define the species. In the second step, you are defining your set of genes or variants by applying filters. So these filters can be a region in the genome, a list of IDs, a particular function, or a phenotype. So this is going to be your input, and you can build simple queries by applying a single filter only. Or you can build more complex queries by applying multiple filters, just like in this picture here. So for example, let's say you have a particular region in the genome you're interested in, but you only want to extract certain genes that have a certain function in that region. So applying these different filters narrows down your search, leaving you with a list of genes with common features. And this is what we apply attributes to in the third step. In this step, you are defining the type of data that you want for that set. So that you want for your results. You are selecting the attributes of your data output. So for example, you can choose if your output should have things listed like IDs, features, variants, if you want the sequences, or if you want any homologs to show on your data table, in the results. And finally, once you're happy with your query, you can view and download your results. You can get these in different formats. So you can get this in HTML, tab separated, comma separated, or Excel formats. Just beware, Excel likes to change gene names into dates. So when you do export it in Excel, just make sure you check the data table afterwards. And finally, for sequences, this you can export in the FASTA format. If you are an R user, there is also a BioMart package available, but this is not developed by Ensembl, but rather the bioconductor community. I've added these slides anyway, in case you're interested. So here are just a few links to documentation and how to install the package on R. And finally, if you would like to know more about BioMart on Ensembl, I highly recommend these 2 publications, which just dive a little deeper into how to use the BioMart tool on Ensembl. Okay, so we're going to move on to the demo now. So using this 4-step system that I just covered, we're going to do a query on BioMart. We're going to look at these 6 human genes, and we want to find out their NCBI IDs, their function, so Go terms, and also extract their complementary DNA or cDNA sequences. So let me just copy the genes to the chat box. And let's just review the 4 steps again, just briefly. So the first step is to select the data set. So in this case, we have genes, and the species we're interested in is human. And then we apply the filters. So we have a set of 6 genes. So this would be our filter. Next, we apply attributes. So what we want in our data table. So in this case, this would be we want to get the NCBI IDs function and the cDNA sequences. And then finally, I'll just show you the different ways to export these results. So with this, I'm going to open a new tab and type in ensembl.org. Okay, so you're quite familiar with this page already. I'm just gonna zoom in a little more just to make it a bit bigger for you. So to get to BioMart directly, we just click this BioMart link at the top of the page on the navigation bar. And that leads us to the tool. So this is what it looks like. So we've got a few options up here. If this left-hand side is populated, if you've done a BioMart query before, just click on new there and it will reset everything. So you'll be seeing the same thing that I'm seeing right now. If you are unsure of how to use BioMart, so if in future you want to do a query and then you're not quite sure how to do that anymore, just click on help and you can find loads of tutorials and documentation on there. So the first step is to choose our data set or database. So as I mentioned earlier, we are interested in genes. So I would click in the drop-down menu here, Ensembl Genes, which will open a new drop-down menu. And this is where we select our species. So we choose our data set. I will click this drop-down menu again. And in the first few species are just commonly used, species that are often used. So this includes human. But if you're looking for your own species, you can just scroll down and look for it there. Luckily, we're just interested in humans, so I will click human genes. And this refreshes the page. So now on the left-hand side, you'll see that we have more options selected. So we've just selected our data set, but now we can also select filters and attributes. So just to make sure we really did select the right data set, it just says here on the left, under dataset, human genes. And so we're happy. We're going to move on to the filters. So I will click the filters link here on the left, and that will open a lot of options here on the right. So the filters are grouped into different categories. So we've got region, gene, phenotype, gene ontology or function, multi-species comparisons, protein domains and families, and variant. And each category, you can click on it. So I would click on region now, and that will open options right underneath. So here you can apply filters that relate to the region. So you can, for example, select a chromosome. So let's just do that. I will click on the checkbox next to chromosome here. And if you want to select a chromosome, you just select whichever chromosome you're interested in. If you want to select multiple ones, you just have to hold down the command or control button on your keyboard, and that will just let you select multiple chromosomes. If you look to the left, you'll see that under filters, it has actually added which chromosomes we selected. So this is just a check to make sure that you really did select the chromosomes you wanted. If you wanted to remove a chromosome, again, if you wanted to just remove one chromosome, you hold down the command or control key and click the chromosome you want to remove. Or if you want to remove all of them, you can just click this check mark next to chromosome, and that will delete all selections you've done there. So you can see under filters, nothing has been selected. So there are a lot more other options that you can choose from in the regions category. So for example, you can also filter by coordinates, so by entering a start and end position. You can filter by multiple regions. So here you use the same coordinate system that we used yesterday when we searched for a location. So it starts off with the chromosome, start position, end position. And then finally, we also have an indication on which strand this would be. So 1 would be for the forward strand and -1 for the reverse strand. So if you have multiple regions you want to enter in this box, you can do that by, you can separate them by comma, just like in this example here, or you just put each position into a new line. Or if you even if you have a text file saved, you can just upload that and you won't have to enter anything on here. I will close this region category now. And I want to show you the phenotype category as well. So if you're interested in a particular phenotype, you can find it here. And the only thing is you can't actually search for the phenotype. So you would have to use your browser search box for that. So to do that, you can enter on your keyboard either Command F or Control F. You probably know this already, but you can search for your phenotype using the browser search box. So for Chrome, the browser search box will pop up on the top right. And I think for Firefox, it will be down here on the bottom left-hand side. Okay, so I'm just going to search for, I don't know, I can't think of anything, cancer. So I typed in cancer in the search box now, and this will highlight all the different phenotypes that have cancer in its name. What you can do is you can scroll down and look for the next highlight, or you can just click this narrow arrow down button, and that will just show, send you to the next word with cancer in it. Just like in the chromosome filter that I covered earlier in the region category, you can select just one phenotype, which will populate this filters section on the left-hand side here. You can select multiple by clicking others, holding down the Command or Control button. Or again, you can remove the filter by unselecting it, which removes it from the filters again. And so there are many, many more filters that you can add. Feel free to play around with them. You can add filters using a list of protein domains. So if you have a list of IDs, you can select the ID type here in the drop-down menu and then enter it here. But for our example, we actually have a list of genes. So I'm going to open this gene category. Oops. And here, again, we have loads of other filters we can apply, but we have a list of gene IDs. So I'm going to search for this section where we can apply a list of genes. Oh, sorry. So that would be the input external references ID list. So that's the second option in the gene category. So I will select this, And I've got the genes copied already, so I'll just paste them. And again here, I'm using a comma-separated list, but you can also enter the list of genes line by line or tab separated, or you can upload the file by selecting choose file just underneath the text box here. An important thing to do here is to actually select the type of gene IDs that we have. So by default, we've got the Ensembl stable ID selected, but these are not Ensembl stable IDs because they don't start with ENS. So I would click this drop-down menu, which will give us all the different options, the different gene ID options. So I know we have gene names here, so that's the 8th option. I would just select that. And that just updates the filters here on the left again. So now that we've applied our filter, we're done with the second step. What I'm going to do is I'm just going to check if we really did apply our filters correctly. So what I'm going to do is I will click this count button on the left-hand side on the top here. And that will just count the number of genes that our current filter applies to in the human gene set. And because we have 6 genes, we are expecting 6 genes here out of 68,016 genes. So I'm happy with that. And I'll move on to the third step now. So the third step is to apply your attributes. So choose what you want to have in your output. I will click the attributes button here on the left. And again, that will open up a new window on the right. And this is, it's grouped in different categories again. So we've got features, structures, homologs, variant, and sequences. So we want NCVI IDs, the function, and the sequences. And to get the sequences, we actually have to get that separately. So we can't get features and sequences at the same time. So I'll just focus on getting the NCVI IDs and the function for now. So you can click these and they will open up subcategories just below. So you can play around with it again during the exercises, but I know we're interested in features, so I will click features. And I know that the IDs we want are external, so I will click on this external category here. So the function, to get the function, we want it in Go terms. So that's the first category here. I will choose to get the accession number and also the name and definition. And as you click these options, the attributes section here on the left gets populated. So I'm just going to remove them again so you can see they do get removed on the left. They also get populated by the order you select things. So if you select accession first, column on your data table, and the results will be added. So that will be the last column. And then you select name, that will be the next column. And then definition will be the next column. And then we're also interested in NCVI IDs. So for that, we go to this subcategory, external references. And here we just search for NCVI gene ID. You can again use your browser search function for that. But I know it's right here. So NCVI gene ID, and that gets added to the attributes again here on the left. So the moment, we have all these attributes. At the moment, all these attributes will be printed on our data table. And we have a list of, we applied a filter of 6 genes, but we don't know which gene has which stable ID. Well, we can find it out, but to make our lives easier, what I'm actually going to do is I'm going to open this gene category, and I'm going to select gene name here so that we can refer our results back to our input. If you wanted to change the order of this, you can unselect everything and then just add the different attributes in the order that you want. So right now, we would have the gene name at the very end of our data table. Okay, so now we've done our attributes. Well, the third step. We're going to get our results. So to get your results, you'll click the results button here on the top left. And that opens up a new window here on the right. So now we have different options. So we can export all our results to a file. We can also get them in a compressed file, or you can get the compressed file sent to your email. So if you wanted to do that, you can just click this and enter your email address just underneath where it says email notification to. And you can also choose your data format. So by default, it's got the tab separated file format selected here, but you can select other formats as well. I'm just going to go click file again. And then underneath, you can see the results, our data table. And by default, Ensembl only shows the first 10 rows. So these would just be the first 10 results of our data table. If you want to see the full table, you can click all. So you select this first drop-down menu, and then you select all, and that will open a new page or a new tab with the entire table of the data that you wanted to get from Ensembl. So this might take a little time because it's quite a big table. Didn't take that long. Okay, so now this is the results that you have. So you can see that each column refers back to the attributes that we selected in the third step. So we've got the gene stable IDs, transcript stable IDs, the Go terms, so what we selected for the function. Then we've also got the NCBI ID, and then in the last column, the gene name. So we know which ID refers to which gene. You might see that there are multiple entries per gene. So for example, here, they all end in the same numbers. And that's because a single gene can have multiple transcripts associated with it, because a single gene can be transcribed to multiple transcripts with a different function. But what you might also see is there are multiple entries for each transcript. And that is because each transcript can have multiple entries of Go terms. So each transcript has all Go terms associated with it will be printed on this table. So that's why you have multiple entries for each gene that you selected. Okay, so I'm just going to close this tab, and I'm going to show you how to get the sequences. And to do that, you can either start a whole new search by clicking new on the top right, on the top left here, or you can just edit the attributes that you had. So I will click on attributes on the left here and edit our attributes that we already have. And I will select sequences and open the sequences category underneath. And here you can choose the type of sequence that you're interested in, that you want to extract. By clicking the different options, it also updates this little diagram. So just as a reminder, which sequences you're actually getting. And we are interested in cDNA sequences, so I will click that here on the right. Again, it updates the diagram. And if you wanted, you can also add upstream and downstream sequences, flanking sequences, but I'm not going to choose that. I'm going to close this category for now, and I will open the header information. So if you see here on the left again, we've got our default attributes, so the gene stable IDs and the transcript stable IDs. We've got the cDNA sequences which I've just selected, but for good measure, I am again adding the gene name because we want to easily find out which sequence belongs to which gene. You can also choose to add other information if you want. So again, feel free to play around with it. So that's all we want. I will click results on the top left again. And this will update the results window here on the right. So you can see it comes out as a FASTA format. And again, it will only show the first 10 rows of the entire data table. You can choose to view them all again. I'm not going to do that because you already know what it looks like, but I just wanted to show you how to extract those sequences. So that's the end of the demonstration. It took a bit longer than expected, but I'll give you maybe 10 minutes to try the exercises in the BioMart tab in the training page. And then I will hand back to Ben for some closing remarks.
Hello, everyone. It's 10 to 5 there in Germany. So I think it's a good time to wrap everything up and just go through the final last things of our workshop before we say goodbye. Firstly, thank you for having us. We hope that you've enjoyed the workshop and that you've found it useful. The first thing that we would actually like you to do right now, if possible, is to complete the feedback survey. So the feedback survey is available through the training pages, through the materials. So if you go to training.ensembl.org and access the training materials as you've done before, you should be able to link for the survey just here. I'll also put. So please do spend some time in the next 5 minutes filling out the feedback survey. We take the feedback really seriously. It helps us to improve our workshops in the future. And there's also an option at the bottom to retrieve a certificate. So if you fill in the survey, and then at the very bottom, you have an option to retrieve an attendance certificate. So if you yes, you'll be prompted to enter your name and email address, and then you should be sent your e-certificate of attendance directly to your email address. So please do spend some time filling that in now. We'd be grateful to hear any feedback that you have about the workshop. So just while you're finishing filling in the feedback survey, there's just a few slides that I'd like to go through just to point you in some future directions if you want to continue using Ensembl. So just to go over what we learned in the workshop today, Ensembl is a genome browser which integrates these four main types of data. So yesterday, Louise spoke to you about the gene annotation data, and I spoke to you about the comparative genomics. And then today we've covered the variation data and the regulation data. And then we finish off by looking at BioMart. So all of this data is available through the website. Obviously, you can then begin thinking about exporting the data on a larger scale through BioMart, like Louise explained. And then you can also access the data on even larger scales through the Perl and the REST APIs. So if you're familiar with programming and writing scripts in different computer programming languages, you can get the data programmatically. And Louise even showed you the FTP site at the very beginning of the workshop yesterday, where you can download the genome-wide file, so for all of the species in Ensembl. If you've enjoyed this workshop, then we'd be really grateful if you recommended us to some of your friends and colleagues at other universities and institutes. We're obviously doing virtual workshops at the moment, and we're happy to deliver those anywhere around the world. And we're also beginning to reinstate our face-to-face training as well. So we'd be happy to talk with anyone who wants to host a workshop at their own institute as well. If you do want to continue using Ensembl, there's a number of ways that you can continue learning about Ensembl and using it. So the first is through the EBI training pages. So Ensembl is part of a larger organization called EBI, and I just want to show you the pages there now. So you may have used the EBI pages before. EBI stands for the European Bioinformatics Institute. And as well as all of the bioinformatics research, we have a number of services, including Ensembl. And if you want all of the other services available, you can click on services in the button in the black bar at the top of the homepage for the EBI website. And here you'll see all of the different tools and databases that we have available. So you can obviously see Ensembl, which is what we've been covering in today's course and yesterday's course. But you can also see all of the other databases. So like UniProt, which has information about protein sequences. PDBE has information about protein structures. Expression Atlas has gene expression data. So there's lots of different databases and tools that all have that work with different biological data types. And they're all freely available and accessible for people to use around the world. As well as our services, we also have training. So if you click on training in the black bar at the top, you'll be able to find information about all of the courses that we have at the EBI. So as well as face-to-face and virtual courses, we also have online courses that you can take in your own time as well. So if you were to type Ensembl into the search bar here, as an example, you can find all of our online tutorials. So you can find any of these courses and take them in your own time. So there's no cost, there's no qualification that you get out at the end. You can take half of the course or just a part of the course that's interesting to you. But it's really just to help you continue learning and developing your skills in using Ensembl and all of the other resources that we have in the EBI. As well as the online courses, we also have a YouTube channel. So if you want to watch some tutorials or recorded webinars that we have about using Ensembl data and tools, you can type Ensembl training into YouTube and find our YouTube channel with all of our tutorials and videos. The final thing is to mention is the help desk. So if you ever have any problems with using Ensembl, or if you have any questions about what you see in the website, then please do not hesitate to contact us using the email address here. So helpdesk@ensembl.org. That will create a ticket in our help desk system, and we'll always get back to you trying to help you understand what you're looking at, or find some data that you're not sure where it might be located on the website. So we can always help you with anything that you might want to find out about Ensembl. You can also follow us on social media. So we've got Facebook pages and Twitter, a Twitter account that where we post information about Ensembl. And then we also have a blog, which is, you can find that on www.ensembl.info. And here we post lots of different things about the Ensembl releases and the new data that's coming, any announcements about downtime that might be coming up if we're performing some maintenance on the website, or job adverts, all these sorts of things we put into the blog. So if you're using Ensembl a lot, it's a really good idea to follow that blog. If you want more information about, if you use Ensembl, we really appreciate it if you cite us. So we obviously have a paper in NAR every year with our annual updates. And then we have some more specific papers as well about different tools and data types as well. So while Ensembl is completely free, we do appreciate it when people cite up the Ensembl papers, because it helps us know who's using Ensembl and what sort of work they're doing with our data. It helps us develop Ensembl in the future. So this is the Ensembl team. I'm not sure if you're expecting more or less people, considering like the size of the data, but it's around about 100 people. And I always get to say at this point, Louise and I have obviously been presenting Ensembl saying, we did this and we did that and we developed this tool, etc. But in fact, we didn't actually do any of it. It's the hard work of all of the people that you see in the picture here. This was taken at the end of 2019, just before we knew what was coming in 2020. We should have maybe taken more of an opportunity to enjoy our time together at them. And then this is just the final slide. So this is our acknowledgement. So this is a list of everyone that you'd have seen in the photo. And then underneath is a list of all of our funders. So obviously all of the infrastructure and the people that make Ensembl possible is not free. So we rely on people giving, or we rely on organizations such as the Wellcome Trust, EMBL and other sources of funding to make sure that Ensembl is free and up to date and available for everyone around the world. So that brings me to the end of the workshop. I'm not sure if Louise, if you have anything else to add, but apart from to say thank you for joining us, and we hope that you found the course useful. Please do get in touch on the help desk if you have any questions. We'll leave the call open for just a few more minutes in case there are any more questions. But if not, then please feel free to go, and hopefully we'll see you soon.
Yeah, I just wanted to say thank you everyone as well, and thank you for all the questions. We hope you get to use Ensembl in the future, and if you have questions, just let us know and we'll get back to you as soon as possible.